everyone. I would like to thank the Scleroderma Foundation, Dr. Varga and Dr. Hinchcliffe for inviting me. It's a great honor. And I'm really grateful for the first good night's sleep in the last 3 weeks. So I always thought internship was hard until I became a father. The first one I was younger, and at least there was only one crying individual. Now it's the second one. So the other day, my older son, who's 4, came to our room and said, "Dad, I can't go to school." I said, "Why? Why aren't you going to school, Ryan?" He said, "My brother was too loud. I'm too sleepy." So my topic is genetics. We are advancing the slides. So the question is, and that's always the million-dollar question, when you have a disease, is it only genetics, or is it only environment, or is it a mixture of both? I think meanwhile we know that all human diseases have a genetic basis to some extent, even infections. When you are exposed to a bacteria, not all individuals get that infection. So your genetic predisposition, the host, is a determinant whether a bacteria, something that's environmental, causes an infection or not. So some diseases are more genetic, some diseases are less genetic. But I think meanwhile we know that all human diseases to some extent have a genetic basis. So anybody knows who is this individual? Any takers? Russian Tsar. So his name is Alexis. He was supposed to inherit the Russian throne, and that's his family. And that is the last Russian Tsar in the middle. And this is the only boy, so the only heir. But the problem was, since he was born, he was bleeding. He was bleeding a lot. His mother was very worried about him. And he survived the bleeding disorder. He was killed 1 year after the Russian Revolution, 1980, because he belonged to the royal family. But his disorder is a disorder called royal disease, and it's a hemophilia. But now we know it's not the more common hemophilia is A, it's actually hemophilia B, because they uncovered his bones, did DNA testing on his bones, and they have pinpointed the mutation that he had. So the story here is, and that's a prime example of a purely genetic disease, where changes, alterations in one gene lead to disorder. So the story behind this is Queen Victoria had most probably, Queen Victoria of England and Scotland, had a mutation, de novo in her genome, in her, as Dr. Hinchcliffe would call it, in her recipe. That was not present prior to that, because that disease didn't show up in that family prior to Queen Victoria. She passed on the genes to one of the sons that had the disease, but this disease is on an X chromosome. In order for the females to get it, they would need to have that alteration on both X chromosomes, which is less likely because it's a rare disease. But because males have only one X chromosome, if they get it, they develop the disease. So that's why the females are usually the carriers. They don't have the disease. They only pass it on, and the males get the disease. And at that time, it was a deadly disease. And interestingly, it affected several monarchies because Prince Albert and Queen Victoria had so many kids, and they basically married it off to different monarchies to increase their influence. They married to the Spanish kings. There was hemophilia among Spanish kings. They married it to Prussian kings because actually Albert was German. And they had also hemophilia. And the most tragic case is Alexis, who would have inherited the entire Russian throne. So basically, the classic example of monogenic diseases. Alteration in one gene leads to the disease. So the question is scleroderma a monogenic disease? The short answer is no. So scleroderma is actually a polygenic disease. So in monogenic diseases, you have alteration in one gene leading to disease. In complex polygenic diseases, most human diseases are complex polygenic diseases, be it myocardial infarction, be it high levels of cholesterol. Predisposition to the disease is caused by several genes, but each gene contributes a relatively small effect, just a little bit. But you put them all together, it creates a susceptible host. It creates a susceptible host that gets a second hit, an environmental hit, or multiple second hits, multiple environmental hits, and develops the disease. So my lecture focuses on genetics of scleroderma. I'm really happy not to be given the task to talk on the environmental triggers of scleroderma, which we know very little about, and it will be a very difficult lecture to do. So what causes scleroderma? Is it nature? Is it just your recipe, your DNA material? Or is it nurture? Is it exposure to some viruses, some food, environment? The answer is yes and yes. So it's a mixture of both. So in order to understand the familial occurrence of scleroderma, we can first look at the disease that is the sister disease, which is lupus. The familial occurrence in the first-degree relatives in lupus is 5%. So each lupus patient has 5% risk to have that disease to reoccur in his or her first-degree relatives. So basically parents, siblings, or offspring. In American studies done in early 2000s, the familial risk was 1.6%, which means, for scleroderma, which means 98.4% of scleroderma patients will not have a first-degree relative that is affected by disease. It might sound little, like 1.6, and it is, thank God, not very high. But this is the most, that's the strongest known risk factor for having scleroderma. There's no other stronger risk factor than having one family member who has scleroderma. So obviously, this add-on does not prove the genetic basis, because obviously your first-degree relatives also exposed to the same environmental exposures. If there is some thinking about adult exposure to environmental triggers, husband and wife pairs that are affected by the diseases are exceedingly rare. There are only 2 cases in the literature. So if this is not purely genetic and it's environmental, that environmental exposure has to occur early in life. Otherwise, we should have seen more husband and wife pairs that are affected by the disease. So we talked about multi-case families. Multi-case scleroderma families happen in 2 different ways. Either you have families that have family members with scleroderma, or you have families where one family member has scleroderma and the other family member has another autoimmune disease. Let's say one family member has scleroderma, the other family member has lupus. So these are the multi-case scleroderma families. And among 1,000 scleroderma cases, we had identified 17 scleroderma multi-case families. And that's an example of that family where round is female, square is male. It's a very unusual family because they had 2 brothers affected by the disease. Usually it's 2 sisters. As you can see, the antibody profile looked also similar. What we see a lot is multi-autoimmune families. Often when I take history, I rarely encounter the situation where they say, my sister or mother has a scleroderma, but I often encounter the situation where they say, my cousin has lupus, my cousin has MS. So this is a family where the mother has scleroderma, and that's the daughter. But also there is a uncle who had lupus, and their cousins who had lupus and MS. So, and I will show you some data that would explain this. So, in order to give a genetic talk, I have to talk a little bit about genetics in general terms, not only related to scleroderma. So, as you remember from your biology classes, we have 46 chromosomes. 2 of them are the sex chromosome, the XY in males and XX in females. But the Y chromosome is the shortest and hardly has any genes. The length of the chromosome decides whether it gets the number 1 or 2. So basically, number 1 is the longest, number 2 is a little bit shorter, and then goes back down there, up to 22. And what you can see, from each recipe, you have 2 copies. So either they are identical or they are different, but each of them is duplicated. And except for the sex chromosome in males, which is not duplicated. In terms of autoimmune diseases, this guy is of special interest. It's called chromosome 6. Harbors a region called HLA region. It's the genetic basis for recognizing self versus foreign. So basically, your body, when the immune system is encountering a molecule, has to decide, is it from my own body or is it coming from outside? If it is from outside, the signal is, let's attack. Let's get rid of it. If it's from the internal system, if it is a wise and functional immune system, it will say, don't attack. You don't want to attack your own body. In autoimmunity, it's not that our immune system is too low or non-functional. It's actually, the immune system is intact. The problem is the recognition of self is impaired. So the body sometimes attacks certain self cells, be it the skin, be it in the lung, and recognizes it as foreign. And the determinant of that recognition sits on this chromosome, which is the HLA chromosome. Highly polymorphogenic, so it's because it has to create so many variations to recognize so many different proteins that come from outside. So you remember the double helix. That's one of the chromosomes. It's not the double chromosomes. It's just the one of these guys. It's basically one here. Looks like that. It's a double helix. And each strand is put together out of 4 bases. And the entire recipe of your body is made up of those 4 bases. It's an amazing system if you think about it. And if there is one base that is changed, look, here is orange, then it becomes here purple, blue, and we color blind. This is blue. That is a change in one base pair. That's called SNP or polymorphism, single nucleotide polymorphism. Most of our studies, the vast majority of studies, examine that type of alterations when there is one change in the base pair. For example, the hemophilia B, remember that we talked about the disease that the young Alexei had it. He had a mutation in a gene called B9, and there's only just one base pair that is off, that caused the whole disease. Sickle cell anemia is another example of that. So most of our GWAS studies examine that type of alteration, but there's also other types of alterations that are not examined by GWAS, would be examined by sequencing. Dr. Varga is heading a sequencing project for Scleroderma and GWAS that would answer those type of questions. Is basically when the whole base pairs are kicked out. Do you see the deletion? And then you have, sometimes there are base pairs that are inserted, and that's insertion. And sometimes you have translocation. One piece is translocated to that. One piece of chromosome is translocated to another piece of chromosome. You see that in inherited cases of trisomy 21. Or there are also translocations in leukemia. So, what are the genes or gene variations responsible for SSC heritability? So, the first study that was conducted was a large genome-wide study with involvement of 10 US centers, including Northwestern University. Northwestern University gives us hundreds of samples. We are indebted to this Northwestern Scleroderma program because it was instrumental in getting that research started. Canadian Scleroderma Research Group gave us samples, and multiple European researchers. By the way, that's a prime example where you need government funding, NIH-funded research, but also you need investigators that trust each other and work with each other to get those type of studies started and to make discoveries that can be followed up with the mechanistic studies. Because none of the centers had enough patients at all. You need really thousands of patients, and scleroderma is a rare disease. So what we call this is Manhattan blood, because it looks like a skyscraper in Manhattan blood. Manhattan, but it could be also called Chicago blood, because Chicago has tons of skyscrapers. And the tower that you see here, the smear, is chromosome 6, that's the HLA region. That proves, because actually in the 1990s, there were people, including some very prominent scleroderma researchers, that were saying scleroderma is not an autoimmune disease, because it doesn't respond well to steroids. And actually it doesn't respond as good to immunosuppression like other autoimmune diseases. But this clearly proves scleroderma is an autoimmune disease. And then there are other hits. That's a T-cell receptor. That is also a molecule that passes on signals also involved in the immune system, but also in other pathways. This is an interferon innate immunity gene. So we discovered some genes that were significant. The way this is put together, these numbers goes from 1 to 22 at the X chromosome. And as you can see, 1 has more single nucleotide polymorphism because it's a larger gene. So it decreases in size. And the Y chromosome is not investigated because it hardly has any genes. Has the gene for hair in the ears and all that stuff, except for it's not much there. These are the hits. And I'm not going to go through them one by one, but I would like to make some points there. So scleroderma is a female predominant disease. So we would have expected tons of hits on X chromosome. We didn't encounter them. So there is one hit on X chromosome. It's also an immune-related logos. It's interesting, but that's the only one that has been proven so far. It might be that it's much more complicated than just looking at the recipe. It might be also important to look how the recipe is folded. So it might be that also epigenetics play a role, which is basically how the DNA is packaged. So I'm not going to talk about that topic because it's not the topic that I was assigned to, and it's also complicated. But it might be that the female predominance of scleroderma is not only in the recipe, it might be in the folding, might be the hormonal influences on the immune system. So this is not the topic that I will be talking about. But the second thing I would like to point out is there are HLA hits on the chromosome 6, remember we talked about that. But they often go with antibodies, and I will talk about that also on the follow-up slides. And then there are all those genes, and the vast majority are immune system genes. But the first one is a T cell. The other TNI1 and P1 can be, is actually I, that can be actually also fibrotic pathways, but also involved in the immune system. That's, for example, is a B cell gene. That's an interferon, innate immunity, B cell. This is a very interesting gene that Dr. Varga has recently published, and we will talk about that. Again, these are cytokines. The vast majority of them, so that's the one that Dr. Varga has published on. This is a immune gene. So the vast majority of the hits are in the immune system. And but you know that scleroderma is not a purely immune disease. We see a lot of fibrosis. We see a lot of vascular involvement. So it's really a puzzle. How do we connect those hits in the immune system to the other components of scleroderma, which is fibrosis, vascular involvement. So a good example of how that connection might work is actually a paper that Dr. Varga and Dr. Bhattacharya just published, I think this month, looking at the link between A20, which is basically based on the alteration in the gene that was discovered in scleroderma is low. And what they show is when the A20 is low, you have activation of the fibrotic pathways in the TGF-beta pathway. That's, for example, one of the few links that we have shown between the immune system and the fibrosis. But we have to do so much more to understand basically how those gene alterations lead to a disease that is fibrotic, that has a vascular component, but obviously has also an immune system. So we have also looked at autoimmune diseases in the first-degree relatives of scleroderma patients. We looked at the 4,600 first-degree relatives of scleroderma patients and asked them, what other diseases do you have? And when we look at that, we see the most common autoimmune diseases in the scleroderma families is thyroid disease, is hypothyroidism, followed by rheumatoid arthritis, hyperthyroidism, and lupus. So it's not a surprise that the thyroid disease is the first hit, because that's also the most common autoimmune disease in the general population. But when we compare those frequencies to the general population, lupus is so much more common in scleroderma families than in the general population. It's 17 times more common. So the answer is here. Basically, when you look at the hits that lupus genetic studies had, and these are the hits that scleroderma genetic studies had, the vast majorities of them are common, including the one that Dr. Varga just published on. That's also lupus susceptibility gene. So something else, most probably epigenetics or an environmental trigger, in a susceptible host to autoimmunity decides whether you get lupus or scleroderma. And we really don't understand what is that something else. So that brings us to another theory that basically immune cells produce naturally certain cells that target our own body, but then they have some filtering mechanism to get rid of those cells. We don't want the cells that attack our own body. We want the cells that attack the microbes. But we think those genetic hits, those genes cumulatively impair that system, make that filter function porous, that few cells slip through that filter and start attacking our own body and create autoimmunity. And that creates the common basis for autoimmune diseases. So we talk in summary that the majority of persons with scleroderma have also antibodies. Sorry, I'm switching gears actually, not so much. I'm switching gears to antibodies because also that's also important for understanding the genetic basis of scleroderma. So the vast majority of scleroderma patients have antibodies that are specific to scleroderma, that only occur in scleroderma. And some of them are more common antibodies, some of them are less common antibodies. Some of them are easily measurable in a commercial lab, some of them are not that easily measurable in a commercial lab because it's not available. But the three most common ones are anti-CL7 or anti-topoisomerase, anti-centromere antibodies, and anti-RNA polymerase 3. Those antibodies can be measured in commercial labs. Usually, actually, I tell my patients what antibody they have. I write it down on a piece of paper and give it to them. Say, this is important because you need to tell other doctors that you have that antibody. Because it's also so far the best biomarker for predicting disease outcome that we have. And the question is, do genetics play a role in determining what type of antibody occur in an individual patient? So this is also a study that we conducted in 2010, where we looked at antibody profile of all cases, and then 1,000 first-degree relatives of scleroderma patients. Then we also looked at controls that were totally unrelated, and we looked also at spouses, because I was interested to know whether the spouses didn't have the disease, but do they have the antibodies that relate to scleroderma? Perhaps they develop the antibodies and 20 years down the road they don't get the disease. And interestingly, the two antibodies that are of special interest is the centromere and the topo1, which is the SCL7. Look at the SCL7. 20% of patients have it. None of the first-degree relatives. None out of 1,000. None of the controls and none of the spouses. There is a caveat to this. This method is done with immunodiffusion. It's the gold standard method. Was done in our own lab. Unfortunately, Quest is using now a different method that is giving a lot of false positives. So if done with the gold standard method, these antibodies are highly specific. Even in centromere, so 291 patients, out of 27% of patients had it. Only two of the first-degree relatives had it, and we called those two patients. At that time, those two first-degree relatives did not have any signs of scleroderma. And then none of the controls and none of the spouses. So that tells me these antibodies are specific. And then the next question is there a familial basis for that? So the first study that was implying some familial basis for that is actually my fellowship, that's the time I was in fellowship, project where we looked at the 18 scleroderma multicase families, and there was no significance between the observed and expected rates of disease type. Basically, it was if your sister had limited disease, and it was not given that you get also limited disease. But when we looked at the antibodies showed a much higher rate of concordance. So if you had a centromere antibody, it was much more likely that your first-degree relative that is affected by disease has also had a centromere antibody. So which tells me there is a common genetic basis for the antibody formation. This is a follow-up study on the GWAS. Then we look at so yellow is centromere, red is topoisomerase or CL7. So when we look at the non-HLA genes, they all go together. So the non-HLA genes, regardless of your antibody profile, look like they are the same direction. But look at that example here. That's an HLA region hit. So this is for topoisomerase. This region represents the risk for topoisomerase. Whereas it does nothing, then you go to the yellow part, you have to go down, like laser is so basically the yellow part, and there's no risk for centromere patients. So the HLA region is a major determinant what kind of antibody you develop. So another puzzle in scleroderma, so these are all puzzles, that's why we need to do more research in scleroderma. We really don't understand how antibodies lead to the disease. Those super specific antibodies, I mean, this is really impressive. When you look at the SCL7, thousands of scleroderma, thousands of first-degree relatives, none of them had the antibodies. These antibodies are super specific, rarely overlap. To this day, we don't understand whether they're just sitting there, they're epiphenomena, or they're truly leading to the disease. So in summary, scleroderma-related antibodies are specific for the disease. There is a genetic basis for occurrence of these antibodies, and the HLA region of chromosome 6 is the main genetic basis for antibody occurrence. And as a summary of the whole talk, scleroderma, the other name for it is systemic sclerosis, is a polygenic disease where alterations in several genes create susceptibility to disease, with each gene having only a low to modest effect. The genetic basis of disease interacts with some yet unknown environmental triggers to cause the disease. Familial occurrence is relatively rare, 1.6%, but represents the highest yet identified risk for the disease. And genetics, especially genes in the HLA regions, also determines of antibody production in scleroderma. I took out this slide that was for my collaborators because the slide I had was for my RNA work, and I realized I'm doing a DNA work, I'm doing a recipe work. So, but I would like to point out that all the DNA work would not have been possible without the involvement of almost all scleroderma centers in US, in Canada, and in Europe. This is truly a collaborative effort. And these are my funding sources, including the Scleroderma Foundation, which has been very generous to our research program. I'm taking questions now.